Good afternoon and welcome. I'm Kirsty Irvin and I'm the new chair of the participant panel at Genomics England. And I'm delighted to be your host today. I've waited a long time to be able to say those words. Please note that this seminar is recorded and will be available on the Genomics England YouTube channel afterwards. Now, today's seminar is 1 hour. Each session is 20 minutes, with then 5 minutes set aside for Q&A after that session. That's going to be hosted by Annalisa. During the talks, please use the dedicated Q&A function within Zoom, or you can do it old school, wait till the end of the presentation and then raise your hand and ask a question there for a verbal question. So moving through, just to briefly introduce our speakers today. So a huge vote of thanks to Sam Tallman for stepping in at short order. I look forward to hearing what he has to say about the impact of the lack of genetic diversity. And welcome too to Alexander Bracanovic. As a parent of 2 preterm babies, I'm really looking forward to hear about the work you've been doing. So a final reminder for the next seminar that's being held later this month, and we hope to see you then as well. So over to Sam. I'll just stop sharing. She says.
Oh, thank you. Let's see.
I don't know if that stopped. Yep, great.
Oh, let's see. Is this, can you see anything, anybody?
Yep, we can see the slides, Sam.
Right, let's play from start. Okay, great. Can you see now everything?
Perfect.
Nice one. All right. Yeah, sorry, this may be a little bit scuffed because it's a little bit short notice, but luckily I've done a few presentations on this before, so we'll see how it goes. So yeah, so I'm Sam. I work for Genomics England. I'm part of the Diverse Data Initiative. So I kind of do a lot of research um, on the impact of diversity, missing diversity, and how do we describe ancestry, etc., and how this may impact things, especially for rare disease. So yeah, happy to share my work on this topic. So just a little bit of background first on like diversity and why it may be important in rare disease for interpretation of variation that we see in a participant or patient. So in the vast majority of cases, we expect penetrant disease-causing variation to be rare everywhere, right? The disease is rare, but also kind of like based on what we understand about humans and how and selection, etc. You expect that there would be less that basically anything that may have a large outsized effect on on a phenotype or on something that we expect these to be very rare, irrespective of where we look in the world, basically. And this is how we kind of prioritize variants and in part how we clinically interpret them, right? So if we observe a variant to be kind of rare in a database or unobserved or that's kind of evidence towards the variant maybe doing something of note or being kind of potentially the cause of a disease. And so, yeah, things like clinical applications, you may look in these databases to see what's going on. Oh, however, this is kind of challenging because over many generations of human evolution, something called population structure has kind of emerged, which is basically just the systematic difference in the frequency of a given variant depending on where you look. And this structure happens at all degrees of granularity, right? You've got big differences between large populations, and in reality, this is kind of continuous across space. Like where you look, there'll be differences. So it's not really the big populations, but it's just the differences in where you look. And depending on so you imagine here, you can see, for example, a mutation starts at this frequency, and there's this kind of separation over the generations. At one point, this mutation may disappear, and another point it may be quite high. And so in reality, that means that like what how rare a variant really is will depend on it may depend on where you look. And it's important to say that most human populations of places, most stuff have quite little differentiation, but there is cases whereby a variant could be quite common or very common somewhere and rare elsewhere. This is by no means the norm, but there is cases in where this happens, and this has been kind of implicated many times, famously in things like false positive diagnoses. So a variant may have been called to be pathogenic somewhere, and in reality, it's actually not. Because when we get more diverse data, we see that it actually is more frequent, and this influences how we then kind of classify a variant. So a variant we could previously thought was pathogenic may not be. And so this has kind of motivated this kind of increased numbers of individuals that we maybe have. So for example, here is 1,000 genomes back in the ancient times of 2015, where we have maybe thousands of people. And over time, kind of you can see that these databases have gotten bigger and bigger towards thousands, and now into the kind of millions of people with these large exome sequencing data sets. But there is still challenges, even despite this kind of enormity of data that we see. One of the kind of well-established challenges is that like a lot of the data we have comes from Europeans or individuals with kind of ancestry similar to people who live in Europe. And this is kind of a common challenge across all kind of forms of data, like GWAS, for example, but also is obviously influential for this particular problem. There's also kind of more nuanced problems. For example, what about structure within kind of these continental groupings? If we use a grouping and we use a frequency to summarize variation within that group, it kind of like minimizes the differences within that group. And in some cases, that can be quite significant. Here you can see individual populations within Africa and the amount of novel variation observed in those populations compared to nomads. So you can see that in some places in Botswana, you may have multiple times more novel variation, and this is related to kind of complex structure and diversity and deep lineages within Africa, and it's kind of another challenge, as well as kind of what is other, right? This is a category which is just people that fit these categories, and also categories that may just not be represented, have a very limited amount of data in from some regions like Oceania, etc. In fact, we can see like false positive diagnosis happening in these underrepresented regions as recently as 2024, right? When we have a little look at a small amount of data, we can see that previously rare variant may actually be quite common. So I want to kind of talk about work that I wanted to do on that we did on the 100,000 Genome Project. So just a little bit of background. I'm sure most people are aware here about the kind of 100,000 Genomes Project. It's a huge kind of British initiative with a kind of large, one of the largest sequencing studies for rare diseases. Many thousands of participants, including many different rare diseases that were undiagnosed prior to recruitment, proband's family members, individuals. And since then, there's been thousands of diagnoses uncovered, many in children. And this has kind of laid the foundation for these kind of routine clinical care, right? So this is an important kind of study, which has formed the backbone of how we now kind of look for potentially diagnostic variation in rare disease in the NHS. Right. And so over time, back in the day, you used to have kind of these early versions of looking for variation, and now we've kind of got to the point where we have millions of variants per individual that we're having to look for, and this is kind of what the 100,000 Genomes Project was able to do. We can capture the majority of at least these small variants and kind of hopefully increase diagnostic yield, which is what was shown. But when you have all of this variation, this massive amount of variation, how do you kind of how do you can't expect a clinician to kind of read the tea leaves of every single piece of DNA. So what you actually need to do is kind of take this DNA and actually kind of prioritize what variation may be sent for clinical review, right? And so this is kind of variant prioritization. I want to kind of like walk through kind of steps or the kind of checks that you may go through to subset these millions of variants down to maybe a couple of hundred or even one, in the greatest case, just with one great, good looking variant. So you start with all of these small variants, and you kind of ask some questions like, is the variant rare? So obviously that's a kind of key question. It's altered protein structure, etc. Um, is it segregating appropriately depending on the kind of modes of inheritance, or whether the disease patterns follows the inheritance patterns within a family, if family data is available, and if we assume that the complete penetrance, which is just related to whether we would expect any variant that may be present somewhere to not cause disease in that individual. That's kind of a bit outside the point, but and obviously with rarity, we're using these large databases like Nomad to say like, is the variant below, say like in less than 1 in 1,000, 1 in 2,000 people or something like that, because otherwise maybe it's just too common to cause a disease. Now it's worth noting that kind of does depending on the disease specifically and other complex things, but in general we're going to be kind of looking at variants that are generally rare wherever you look. So if not, you may filter those variants out, just so we're not even going to bother with those. And then, we have these kind of big, great evidence bases of disease associations. And maybe the next kind of step will be, well, okay, the variant fits all of those categories. And the next tier of priority up, you may want to kind of you may want to see whether the variant, it fits what we expect if it's going to be a causal variant within a database like ClinVar. And then you have these kind of variants that are sent for primary and secondary review, right? So maybe we really want to review these, and then maybe afterwards you would want to review these on the basis of new evidence or something like that in GEL, in Genomics England. This is at least kind of how we still do. There's other steps in bits and pieces, and these obviously are developing. This is kind of the broad kind of backbone of this small variant prioritization process. So how does this kind of look if we think about participants in the 100K? How does this kind of look, and how does maybe ancestry influence this? Well, I talked about population structure and kind of these systematic differences in allele frequencies. Well, one of the ways of visualizing this is to kind of imagine to kind of collapse this complex set of frequencies into kind of like lower dimensional space using PCA. As you can see, this kind of scatter plot of differences, if you cluster closely together, you generally your frequencies of the mutations that you share are more similar. And you can see that this kind of corresponds to geographic locations of population of people. So you can kind of say this individual is genetically quite similar to people who live in Africa. And we kind of talk about that as being like inferring where your ancestors may live, or where your ancestors lived in the past, or who, but in reality, it's just who you're similar to in these databases, right? So you can see the majority of people probably that's expected in a cohort. You have the majority of people look similar to people who live in Europe with other differences. But you can see that there's still quite a lot of structure within these classifications, and that was obviously something that I was mentioning before is a kind of thing that I'm particularly interested in. And so in reality, we can actually kind of go further and say, well, if we compare to a smaller subsets of individuals, maybe we can see kind of patterns that we otherwise would have missed, right? So you can start subsetting individuals down into groupings that are more granular, maybe like if your ancestries are from East, it's more similar to people who are living in Eastern Africa or Western Africa, for example. So if we look at all of the kind of these candidate protein altering variants, all of these variants that are prioritized across the 100K, we can see the vast majority of them are rare everywhere. There's a minority that are a bit more common that are under recessive disorders, but this is a small minority. And you can see that this is the kind of just distribution of how many we find and how many were kind of sent for these review stages. And these kind of just correspond largely, at least these peaks, to the availability of family data. This is the kind of key differentiator, and that's just because if you have something that's segregating, if you don't have family data, you can't apply that filter at all, right? So this is like just to kind of keep in mind, that's the kind of primary thing that's going to cause any big differences. But if you actually look and kind of try and stratify and correct for all of these different other variables and say, well, do we see a difference, an association between these groupings, these people to whom you're similar with, to and your numbers of these variants that are being sent for review, you can see obviously there is significant differences across everywhere, right? Even across all of these categories. And some of them, some obviously more than others. For example, Eastern Africans, this is compared to the majority kind of Northwest European group, which is like the majority of people in the UK, similar to people in Northwest Europe. You can see that this East Africa group might have up to three times more kind of variants that are being sent for review, right? And so if you look at kind of, you can kind of get some clues as to what may be happening here, right? So you can see actually that if you just look at the differences here and you kind of correlate this with the differences in the number of variants that are just absent from Nomad at all, you can see the correlation is pretty clear. So this is clearly being largely driven by the relationship between the participant and the people in the databases we're using to determine whether a variant is rare, right? So if you have more close relative people whom you're kind of more closely genetically to in these databases, you're going to have differences. If there's fewer of those individuals, it's going to be more and more difficult to know whether a variant is actually rare, right? And I'll kind of go into a bit more detail about that as well. You can actually see more evidence of this if you look at things like predicted functional impacts, right? So you expect, as I said at the beginning, a variant with a kind of deleterious functional impact should be rare everywhere. Whereas maybe if you are prioritizing more common variation, you expect there to be a kind of lower proportion, and this is actually what we see. So individuals that are having more variants prioritized, a lower proportion of those variants are predicted to have functionally damaging impacts across different effect predictors. So this is kind of evidence that you're probably getting stuff that's probably more common being prioritized in individuals who have less kind of similarity to the people in these big databases. And we can see kind of more evidence for this if we use these kind of groupings and we look at a different cohort. And so from the UK. So this is from the COVID-19 cohort at Genomics England. And we can kind of ask like, well, okay, these variants that are rare, how many of them would be, how many of them kind of are quite likely to be above this frequency threshold in one of these groups? And you can see that in the this group. So you can actually see that there's tens of thousands of these kind of candidate variants that are rare across all of these different groups in Nomad, but are likely to be above this frequency. This is kind of a statistical correction for differences in sample sizes. Obviously, you can't tell with a group of 100 people how rare, one in 1,000, obviously, but you can kind of tweak it at least. And you can see that like there's the majority of these are appearing in the groups for whom there is lower, similar to the individuals for whom there is more variation, right? So you're basically prioritizing mutations that may not otherwise been prioritized if the composition of the ancestries in the kind of cohort is different. And these include, and this is actually true. You can see actually cases where there's variants that are at least even an order of magnitude more common, or likely an order of magnitude more common as well. And most of these occur in as well in these similar, potentially underrepresented groups. And so I was talking about this for both reviews. You can see similar patterns. Obviously, there's far fewer of these. Maybe just 2% of the variants appear within these panels, but you can see the patterns are reasonably similar. This is true even of the high priority variants sent for clinical review. You still see kind of up to 3 times more of these variants being prioritized, and the patterns are pretty dissimilar. You can see similarities for functional predictions as well. Obviously, this is a little bit more hazy, but the kind of patterns are more similar. You get more panels, you get more kind of stuff that isn't looking like it's got functional impacts being sent. And so now you can look at kind of see what's the impact of that clinically. Well, if you look at these high priority candidate variants, a large number of them have been kind of the basis for all of these pathogenic variants or causal variants that have been identified in the 100K. And you can see actually the kind of similar impact here, right? So like the predictive value, so what fraction of the mutations that were sent for review were accepted to be pathogenic or causal for the disease. And you can see that this kind of similar pattern emerges, right? You get a lower fraction of these, so a higher rate of kind of like false positives. I guess the predictive value is just like how kind of, if you can think of like true false positive signatures, you can see the same pattern emerge when you correct for all of these facts. This is just the raw, and then you can see the correction correlates kind of nicely with differences in just the number of these variants you're sending. And so this actually, yeah, so you can see like only 2.5% of the mutations here, whereas up to, 10% significantly higher in the kind of more well represented groups, or groups of bigger representation in these databases. And in reality, but in reality, if you look at actual yield, so the actual just how likely is it for any one of those mutations to be pathogenic, that there's not really much difference at all, right? So the overall diagnostic yield is not particularly different, just naively looking across the whole cohort, kind of trying to correct for groupings of differences in disease. It's very difficult because the number of diseases and variation here is very complicated, but there's at least no big evidence that like the individuals, any one kind of individual based on their ancestry had differences in the kind of diagnostic yield when you correct for different things. And to kind of like suggest that like and this kind of makes sense on the basis of if a variant is really rare, it will be rare everywhere, right? And so you will still prioritize it. It will just be alongside a lot more other stuff that you don't really know what's going on. And you can actually see that evidence for that like uncertainty if you look at like how many of these gene panel variants are assessed or reported as VUS, right? You can see that actually an even greater difference in the number of these. So you can see, although there's no difference in the actual likelihood of observing a kind of pathogenic variant, there's a lot more variant. We don't really know what's happening. And it end up being evaluated as VUS, probably because there's not enough evidence to kind of actually say that they're not pathogenic, right? So I imagine the vast majority of these differences are benign variants that don't have the evidence weighting to say that they are actually benign, and so they end up being these uncertain classifications. And so yeah, so just some kind of conclusions, I guess. Yeah, it's important to kind of note that if you're seeing a variant that's rare, you need to understand how what that actually means with respect to the participant and the databases you're looking at, right? And you can see this based on the fact that there's this excess of these superficially rare variants in individuals who are kind of less well represented in these large databases, and it's going to increase uncertainty and increase VUS. So obviously we need more data, is a kind of or new kind of ways of thinking about this, to kind of identify some of this missing common variation. And also important, yeah, to kind of emphasize that these groups are kind of statistical constructs, and so you can't kind of assume a finding will translate on the basis of a discrete label like African or whatever. Like there's always going to be some heterogeneity, and kind of structure exists at all levels of granularity, and you kind of have to think about this in a more kind of personalized or continuous approach, which is something that I think is also pretty important. So yeah, thank you very much. That's the presentation. The paper is out on medRxiv and is currently under review at the New England Journal. So hopefully it's not too horrible, but well, yeah, I'm ready to take your questions.
Brilliant. Thanks so much, Sam, and especially also at such short notice. There are a couple of questions already, but please do add questions. So one of them is about the data that you've presented being split, so east, west, north, south for Africa, and somebody wanting to know the source of that data, and also if it has normal control data.
So yeah, so the source of that data actually derives from the UK Biobank, which has a lot more granular insights into the geographic location of individuals. And so you can, it was actually, I wanted to find a database that was kind of a reference set of individuals that were quite at least as similar as possible to people from the UK that live in the UK. Now, the labels and definitions are kind of a bit muddy, and the groupings don't separate always by geography. But generally, the database is publicly available set of kind of reference groups from the curated based on the UK Biobank, and control individuals. I'm not 100% sure what that means in terms of this, but I guess yeah, that was how it was done. And you could the kind of, I guess one of the main points is that you can do there's no true there's no truth in this. You can set your threshold dependent on whatever you want. You more or less people, maybe more or less similar, dependent on how stringent you want to be. Kind of the main, I guess, point is to kind of identify participants, who may be at greater risk of false positives, etc. And the labels are kind of more of an art than a science, in my opinion. Yeah.
Yeah, and I think there's a follow-up one to that before I go back to the first question. I don't know if you can see them written out as well, but it's about the current Nomad release having data sets from UK Biobank that could contain affected individuals. And what were your views in interpreting variants that have now become more common in those databases, bearing in mind that we don't know for sure the health status of the individuals?
That's a really good question. I think that's really specifically, I think that's quite disease specific. And I think that's
Yeah.
another thing that's important to kind of make sure that to kind of emphasize is that diseases and how common a variant is, these kind of approaches for prioritization are obviously quite kind of just they're quite standard. They're just very standardized, and the same people kind of go through the same procedure. But in reality, the kind of how rare you expect a variant to be in any set of individuals will really depend on the specific disease and the specific phenotype you're looking at, right? So there may be extremely kind of severe phenotypes or extremely kind of rare disease where even in the case of the UK Biobank and everyone that you shouldn't observe the pathogenic mutation there, or it would be much more likely to be some high impact de novo alteration where you expect anywhere you look it would be extremely rare. In other cases there's kind of stuff that is almost certainly actually disease related that's common in Nomad and other things like that. These are much rarer cases, but there is circumstances where that happens related to things like low penetrance or related to the fact that the disease may actually be more adult onset, in which case it doesn't really influence your ability to have children. And so, there's a kind of big spectrum of things. And I would say that there's probably no hard and fast rule, which is probably what I would say. But I do think it's interesting. And I think it's useful for Nomad, for example, in those cases. If you are concerned about that, I think they do provide like the ability to at least curate cohorts on the basis of the different groupings and stuff like that. And so I think for extremely high penetrant rare diseases, it should still be much rarer even than the thresholds of 1,000 in the vast majority of cases. But I think this is kind of there's probably no hard and fast rule. I'm not a clinician, so I would, or even a rare disease expert, but that would be my personal kind of view.
Yeah, I think, and obviously the penetrance as well, and we don't actually know the
Yeah,
real penetrance for a lot of rare conditions.
exactly.
So that's another complicating. There's a question about how you ran the population structures and whether you were using common and rare variants to do that.
So no, actually, the kind of the analysis of the population structure is based on sets of kind of high quality common variation. If you use rare variation, it's an interesting question actually, because
whether you get into your circular.
Yeah. Yeah, so no, yeah, so it's that was used based on common variation, and I'm not really looking for really fine scale structure here. Like rare variation will be probably useful for identifying quite fine scale structure, whereas kind of groupings based on common variation, I guess, is a little bit more separate. And also, yeah, but you can use rare variation, and it may do different things. I also looked in things like rare variant sharing and things like that, and obviously you can see a part of that related in the way that I look at these candidate variants. But yeah, I was using common variation as to answer the question. Yeah.
Brilliant. And there's one last question, which is about excluding variants which might be common in particular ancestral groups in Nomad, and asking about a recent paper which included a variant which actually wasn't rare in Africans. I'm not sure I know. Yeah.
So what a variant that wasn't rare. So is this suggesting that there's certain pathogenic variants probably will be common in certain groups? Is that the kind of question, or is that
That's what
basically
that's what I think it relates to, but maybe in the chat, maybe is David Curtis, maybe you could talk in the chat and clarify the question to answer in the chat while we move on to the next speaker. Is that all right?
Sure.
So
Great.
yeah, brilliant. So yeah, thank you so much, Sam. I really enjoyed it, and it was brilliant that you could step in. Thank you.
No worries at all.
Kirsty, shall I hand over to you?
Oh, can I stop sharing? How do I do that? Sorry.
I'm so sorry. Operator error there. I think we were just going to hand straight over to Alex there. You're good to go, Alex?
Yeah, just one moment. Can you see that?
Still waiting, still just see you. There we go. That's great. Thank you.
Okay, thank you very much for the invitation to talk today. I'm just going to be talking about a master's project that I did looking into the challenges of interpreting rare missense variants in women diagnosed with intrahepatic cholestasis of pregnancy using the Genomics England research environment. So just to give a bit of background into ICP. So this is a liver disorder which develops in the late second and early third trimester of pregnancy and is characterized by pruritus, which is a severe itch and increased serum bile acids. And it is quite complex multifactorial disorder with hormonal, genetic, and environmental factors that all play a role, and is quite rare in the general population. Although it's worth mentioning, it is the most common liver disorder in pregnancy, with an incidence of around 7 in 1,000 pregnancies in UK, and more common in South Asian women, about 15 in 1,000 pregnancies. So to talk about the clinical importance of ICP, so as I said earlier, the characteristic symptom is pruritus, and as you can see from the images on the right here, so you have the first image, which is a very early stage of itch, and then a more severe case later on the right, and you can conclude for yourselves that other symptoms such as anxiety and sleep deprivation can come from this disorder, and away from the mother, you also have risks for pregnancy as well. So ICP is associated with preterm birth. About 1 in 10 women diagnosed with ICP will have their baby early. And in general, the risk to the fetus is elevated in women with serum bile acids of a concentration over 40 micromoles per liter. So there is a direct correlation with the increased bile acids and risk of stillbirth and perinatal death. So to delve into the genetic contributions to ICP, we have both rare and common genetic variants involved. So rare mutations in 2 genes in particular, ABCB4 and B11, which are transmembrane proteins, have been reported in 25% of severe cases of early onset ICP, and around 16% of all ICP cases have an ABCB4 mutation. So these 2 genes are quite impactful in terms of disease manifestation. And a similar sort of story is seen with common genetic variants. So this was a genome-wide meta-analysis published by Dixon et al. In 2022. And you can see that ABCB4 and B11 were also found as significant loci impacting disease on the common variant front. So just to go into these 2 loci, but particularly ABCB4, as it was the loci that I investigated in particular in my work. ABCB4 is a is a transporter protein, essentially transporting phospholipids from the liver cells into the bile, and its main function is helping protect bile ducts from toxic bile acids. It's worth noticing that ABCB11 is the bile salt export pump, and its job is exporting bile acids into the bile, and phospholipid secretion of ABCB4 is triggered by bile acid secretion in ABCB11. So these two are very much intertwined and interlinked. Yeah, so I will just briefly run over this. This might be fundamental for many of the researchers in this at this seminar, but essentially looking at small genetic variants, we can broadly categorize them into synonymous and non-synonymous. Synonymous being a change that doesn't cause an amino acid change and therefore easily can be categorized as benign. And then non-synonymous variants can be split into missense and nonsense. Nonsense being the more easier one to categorize as pathogenic because it initiates a premature stop to the protein, and missense variants can be more challenging, and I'll get into this a bit later. Sorry. Just regarding rare missense variants, a minor allele frequency of less than 0.01 would classify a variant as rare, and low frequency is normally less than 0.05. And the general trend is that the lower the frequency, the higher the effect size. So we're expecting in rare variants to see a larger effect size than, let's say, the common variants found in the GWAS that I mentioned earlier. So why can missense variants be challenging to classify? Some missense variants result in normal or just slightly reduced functional consequence, and others result in significant reduction in protein function or its overall stability. And reasons for this include whether or not the change in amino acid results in a significant change in overall structure or stability of the protein. And so a sort of guideline that we have to classify variants has been published in this flagship paper by the American College of Medical Genetics and Genomics, ACMG, which helps classify variants based on 5 categories of evidence: population, computational, functional, and segregation data. And just look into the computational data. So this is a table here that demonstrates some of the advised tools when the paper was published for classifying missense variants. Notably, you have SIFT, polyphen, and CAD, which are very frequently used in variant classification. And of course, this list isn't exhaustive, as I'll come on to later, because constantly new tools are being developed. But the important thing here to notice is that computation tools have quite a large burden on missense rare variant classification. So you can see in this paper, removing evidence supporting likely pathogenic classifications in regards of computational evidence caused 24% of likely pathogenic variants to go into variant of uncertain significance. So they do hold an important role when sort of trying to determine the functional effect of these variants. And in general, the rules for using these tools as evidence as cited by the ACMG are you have to use multiple software programs for sequence variant interpretation. So you can't just use one to make to give you that evidence for computational work. And they all have to agree on either a pathogenic or benign prediction. So if you have some discrepancy between predictors, you can't use that. You can't use computational evidence as a tool for classification, which, as I said before, they hold such a large burden on overall classification. This can be challenging. Then you end up with a lot of variants of uncertain significance in those cases. So just to go over very briefly some of the categories of variant predictors and how they make their predictions. So it's important to note that most of these variant predictors are supervised machine learning algorithms and require training and test data sets. And so you have, for example, homology sequence predictors, which are based on evolutionary conservation and they imply that an amino acid change in conservative regions are more deleterious. So an example of a tool using that would be SIFT. And then you have a combination of sequence structure and evolutionary conservation. So sequence structure based on features of the protein affected, such as polarity. And these are normally accessed through databases such as UniProt. And Polyphen2 is a classic example of one that uses this. And then you've got combinations of conservation and functional information, i.e., the number of variants in a known disease-causing gene. And CAD would be a good example of one that uses that sort of algorithm. And the reason I just go over these is to introduce alpha missense, which is slightly different from the others in that it makes its prediction primarily from its AlphaFold protein structure prediction tool. And so is able to make predictions on general protein structure, but also changes caused by different amino acids, which is obviously quite useful in classifying missense variants. So just going quickly onto our methods, how we selected our cohort. So we use a Genomics England research environment to filter for patients that had reported ICP based on the ICD-10 code, and we found 193 women falling into this, and we then pulled the variants in ABCB4 for all of these women using the GEL's small variant workflow, and filtered to only keep a missense low frequency variants, so less than 0.05. And we found 9 missense variants that were found within this population of 193, and ran 5 in silico tools, including SIFT, Polyphen, and CAD, which I've mentioned earlier. VESA, which is a bespoke prediction tool for classifying ABCB4 variants in particular, and only ABCB4 variants, and then alpha missense also. And so this is just a little topographic demonstration of the protein changes that we found in ABCB4. As you can see, the vast majority of them are located in the nucleotide binding domain. I have a couple in transmembrane domain regions. And you have here a table, a tablified version of that, and also including GenoMAD population frequencies. You can see that they're very low. And then some of the predictions from each of the in silico tools. You have these red boxes indicating deleterious and possibly damaging variants. You have the green ones that are benign and tolerated. And then you have this one ambiguous one, which was offered by alpha missense. So just to highlight the first sort of, let's call the challenge in interpreting these variances, there's evident disagreement between predictors. So you can see that only 4 of 9 variants had full agreement over classification into pathogenic and benign. And to present a kind of resolution for this is the use of ensemble meta predictors. So you can see this study was done looking into false discovery rate. And you can see alpha missense ranks very highly there, but just underneath it is Revel. And Revel is now becoming a very common in silico tool to be used. It weighs the scores of 13 different tools, including SIFT and Polyphen, and produces an overall score based on all of these tools. It is important to note that although it does combine many different tools, the ACMG does state that it recommends the use of multiple different tools. So especially in clinical settings, you would expect this not to be the sole tool to be used in classification. It's just worth mentioning another issue that normally comes up with these tools is circularity. So variants from the same gene are more likely to be classified the same on the basis of prior knowledge of variant effect. And many of these tools are trained on similar data sets. So you have this issue of maybe overinflation of certain variants. The second challenge that we have here is comparison to functional studies. So you can see here I've taken 3 of the missense variants, amino acid changes that we found in our work. And you can see here alpha missense predicts benign, VESA, the bespoke tool, predicts pathogenic, and then an independent functional study concludes no detectable effect. You get the vice versa here in the second variant, and in the third one, although both variants, variant effect predictors agree in a pathogenic classification, it doesn't congrue with functional study data. So it shows a little issue with that, and maybe highlights that lab-based functional assessments are still the gold standard for classifying variant effects. Okay, so the third challenge we can conclude from this is also, why does alpha missense predict differently? So you can see from the table again here that the main difference in this disagreement between predictors is actually that alpha missense in many cases is predicted differently than the bulk of the other in silico tools. And I mean, this can be expected. We know that alpha missense predicts differently, but it prioritizes its AlphaFold database rather than the standard prior knowledge. And so we ran in our dataset a couple of statistics that are commonly used for these tools, so the MCC and F1 statistics. So the MCC is essentially valuing balanced predictive accuracy, so it can only be high if the predictor performs well with both positive and negative values. And F1 performance based on increased weight on determining true positives. So we can see in our data for ABCB4 and ICP that alpha missense performs slightly better for this balanced accuracy. And this has been replicated in many studies before, and suggests that alpha missense can be very powerful in assessing novel unannotated variants. Just to look into some future directions of work that are happening in our group. So looking more into the investigation of overall heritability of ICP, incorporating common and rare variants is something that's being worked on in our group now, since we know that both common and rare variants have an effect on ICP manifestation. And then we have another project, which is GRACE, which would be incorporating researchers who have ICP genetic data into a larger meta-analysis to look further into the common variants that were first published in the 2022 paper. And just to conclude on this work, so one of the first conclusions is that the use of meta predictors such as Revel have shown to have higher prediction accuracy than individual conventional tools, but it's important to note that the single burden on one tool is not obviously preferred in clinical settings, but can be useful in research settings. And the use of alpha missense may be best implemented in variants that are novel or initially classified by other tools, but have resulted in uncertain significant classifications. And in terms of the disease itself, it's important that the most reliable tools are used in classification to provide the greatest clinical benefit to patients. So that's obviously the most important thing. We want tools that are the most accurate to help inform our decisions on patients. Yeah, so thank you for listening. These are some of the acknowledgements, some of my supervisors and other team members that helped with this work. Just stop to share.
Thank you so much. I really enjoyed that, and working in clinical genetics, it's always really nice to hear about different tools being explored and yeah, where you think they might be relevant, and lots of claps and hearts. Interestingly, and you may already know about this, but David Curtis has said in the Q&A that they've done a systematic evaluation of the performance of alpha missense and other predictors to identify variants affecting susceptibility to common disease, and there's the Nature link. So I don't know if you want to comment or...
No, I think that's something I should definitely look into more. Yeah, thank you for that.
I was wondering if a similar thing, if people had this is a naive question, but if people had done similar things for other conditions that you know of, and whether they'd have found similar patterns to what you found.
Actually haven't seen similar things in the way that we've done it in the Genomics England research environment or in other large different biobanks. But for sure, I think that most people who have worked with rare variants have come across this challenge of, you have this mass of data and then you find your rare variants, and then the challenge actually begins at that point where you're trying to interpret them. And it can be very challenging, as you can see with some of these tools, not agreeing with each other. And of course, disagreement in these tools doesn't work in your favor either, because you end up not including computational evidence in classification, which is, you have other evidence bases like segregation data. So having family data, it's not always available, in many of these rare disease cases. So normally, if you don't get that computational evidence, you're at a higher risk of having a variant of uncertain significance, which is not helpful really in clinical or research settings.
Yeah, and I don't know if this is relevant or not, but I don't know if there's a difference with these tools following the ACMG guidelines or a kind of Bayesian probability. So if I have had a patient in the past who had a sort of unusual novel gene change, but it was a really, really specific phenotype. So the ACMG classification came out as a variant of uncertain significance, but on the Bayesian classification, it came out as very highly likely to be pathogenic. And I just wondered, if these tools, if that affects, or whether you just looked at ACMG.
No,
This is just
I
out of
mainly
interest.
focus on ACMG. Yeah.
Yeah, which obviously most people are using. There's a question about how can you elevate the level of accuracy of the in silico prediction for missense variants to meet the lab results?
I think one major issue is this issue of circularity. So the fact that we have very few databases really with actual curated functional data means that even if you include different algorithms, they're essentially all trained on very similar data sets. So I know that there have been suggestions to include phenotype specific databases. And of course, what Alpha Missense have done is great, because they incorporate a completely different database with AlphaFold that's not used with any of the other tools. So all of this sort of diversification of training sets is really important in improving accuracy. So I think we've come in great leaps and bounds with Alpha Missense now. I think it's now worth maybe thinking about, like I said, more specific phenotype developed databases for training sets.
Yeah, interesting. Peter Dixon's also commented about lab assays not necessarily agreeing and having 4 different ones for ABCB4 function. I don't know if you want to add anything there, or
I guess, yeah. So what Peter is saying essentially is that we have very few actual assays for determining ABCB4 functions. So yeah, that's a limitation of those functional assays is actually the very few amount that we actually have to make those functional assessments. So yeah, a bit again the issue of leaning on in silico tools when we're maybe not quite there yet with in terms of the accuracy.
Yeah, and this is not meant to be an interview style question, but is if money and logistics were no object, what would you do next?
Any logistics
if you were free to do whatever you wanted, what would you do next?
Yeah, I would potentially maybe look at how you could implement maybe some of these similar or conventional tools that were initially described in the ACMG, and maybe tailoring them. For example, VESA is a tailored tool for ABCB4 that I mentioned. And it might be worth maybe integrating some of these new data sets into VESA, just to maybe see whether or not that changes, or how much that changes its prediction compared to Alpha Missense. Yeah.
Thank you. I can see somebody's asking about, does Genomics England have the option to recall by genotype for deep phenotyping? And I'll take that one. So it depends is the answer, and I'm happy to for someone to, if anyone wants to email for further discussion, that would probably be the best, because it would depend on specifics. But so if somebody was in, for example, the 100,000 Genomes project, or the has shared their data in the National Genomic Research Library because they were part of the Genomic Medicine Service, depending on the reason that they were having testing in the first place. So if somebody wanted to find patients with particular genotypes and do further deep phenotyping, then we can go back to their clinical teams and say, this is this researcher is interested in this. This is the study they're doing. We're sharing this information in case you'd like to contact them to the researcher to find out more information. So we do these clinical collaboration requests. What becomes ethically more challenging is if it's say parents who were recruited unaffected and somebody wants to select by genotype on a kind of predictive late adult onset condition that people haven't sort of consented to find out their risk for, then that becomes ethically difficult to go back. So it depends, and I'm always happy to discuss specifics. Thank you both immensely. They were fantastic talks, and I'm going to hand back to Kirsty to close our session today. Actually, Kirsty, if you don't mind, I'm just going to plug that we made a podcast for Rare Disease Day, if anybody wants to listen, which was Can Patient Collaboration Shape the Future of Therapies for Rare Conditions? Back to you, Kirsty. Thank you.
Fantastic. Thanks, Annalisa. And thank you so much to Sam and Alex for your informative sessions today. I have to say it's really a privilege as a 100,000 genome participant to see how our donated genetic material is put to use and then the real world impact of that research. I can't, as a solicitor, I have to say I didn't necessarily follow all of the slides, but I'm sure everyone on the call today really enjoyed your insights. So I wish you all the best with your academic careers. May many grants and research fellowships come your way, and we look forward to seeing everyone for the next session on the 25th of March. Thanks so much. Bye-bye.